What up, Bonner Bros? Today we'll be looking at a scientific journal written by the faculty of the University of Dundee. It is titled Retinoic Acid Orchestrates Fibroblast Growth Signaling to Drive Embryonic Stem Cell Differentiation and was conducted by Marius P. Stavridis, Barry J. Collins, and Kate G. Story. Embryonic stem cells are pluripotent stem cells found in the inner cell mass of a blastocyst. This means that they can either self-renew or fully differentiate into any somatic cell type. They are known to be able to propagate without differentiating for an indefinite length of time. This, however, only happens if the conditions obstruct the cells clumping together or an environment in which an unspecialized state is maintained. In mouse embryonic stem cells, in order to move from the dynamic self-renewal stage into differentiation, the signaling of the fibroblast growth factor (ERK) must take place. Proliferation, migration, and differentiation are among the vital processes controlled by fibroblast growth factor during the development of all mammals. It is already known that blocking fibroblast growth factor (ERK) signaling promotes self-renewal in mouse embryonic stem cells, whereas exposure to it is required to differentiate into an adult cell. Retinoic acid is a metabolite of vitamin A and is a known signaling molecule that also promotes differentiation of cells. It is also known to interact with FGF8. In this study, their aim was to investigate the mechanisms by which retinoid signaling promotes mouse embryonic stem cell differentiation. They used a variety of scientific methods to arrive at these conclusions, namely immunoblotting, immunocytochemistry, flow cytometry, and quantitative RT-PCR to demonstrate their findings. Now, to demonstrate these findings, we will see that embryonic stem cells are in a dynamic state of flux where they can either stay as self-renewing or can go on to become somatic cells by differentiation, as seen here. Now, it is already seen that FGF-ERK pathway is needed to progress from this state to a state of differentiation. What this study found is the endogenous retinoid signaling is required as retinoic acid advances differentiation by a dual mechanism. This mechanism causes an increase of fibroblast growth factor signaling, but then an overall decrease of this signaling. This is due to the retinoic acid inducing FGF8 and ERK at day 1. This stops the self-renewal here. Now, the gradual repression of FGF4 causes cell differentiation to advance. Why is this? This is because the cells have already underwent a period of renewal. The inhibition of FGF then has the opposite effect to what it would usually have, and therefore causes differentiation to increase. Therefore, retinoic acid is very important in this process. Thank you for watching the Bonner Show for our Bonner Bros, and always remember, science is cool.